Okay, we're back. Let's talk about synaptic transmission. This is electrochemical actions going across the synaptic cleft. And these can be excitatory or inhibitory actions. Keep in mind that we have these electrochemical messengers, these neurotransmitters, that will increase postsynaptic membrane permeability. In this case, it'll be to sodium ions, and they would trigger an impulse, and thus they would be excitatory. Other neurotransmitters may decrease membrane permeability to sodium ions, reducing the chance that it would reach threshold or reach potential. We term those inhibitory. When an impulse reaches the synaptic knobs of the axon, the synaptic vesicles will release that neurotransmitter into the synaptic cleft, into the gap there. The neurotransmitter then will react with these receptors, these cups on the other side of the postsynaptic membrane. There's more than 100 neurotransmitters being produced by the nervous system. Most of these are synthesized in the cytoplasm of these synaptic knobs, and they're stored in synaptic vesicles. We have several different types. We have some that we term acetylcholine, we have biogenic amines, amino acids, and then there's these 50 neuropeptides, or more than 50 neuropeptides. So these would be, keep in mind what a peptide is. Peptides are what make up proteins. When the action potential that we've talked about reaches the synaptic knob, now we have calcium ions rushing in. We've had sodium, potassium, now we have calcium. They rush in, and in response, some of the synaptic vesicles are going to fuse with the membrane, release their contents into the synaptic cleft. All right, bullet 3 on slide 40, chapter 9. We're talking here about enzymes. Keep in mind that an enzyme is a biological catalyst. What's a catalyst? It regulates the rate at which chemical reactions take place. So we have these enzymes within the synaptic clefts, as well as on the postsynaptic membranes. These rapidly decompose, these enzymes rapidly decompose the neurotransmitters after their release. Chemical reactions here. And then we have a destruction or a total removal of the neurotransmitter that would prevent continuous stimulation of the postsynaptic neuron. All right, slide 41, impulse processing. How impulses are processed is dependent on how the neurons are organized in the brain and in the spinal cord. We have these features termed neuronal pools. All right, neurons that we find within the central system are organized into these neuronal pools, and they have varying numbers of cells. Each pool, each neuronal pool, receives input from afferent nerves coming in and processes the information according to the specifics of that type of neuronal pool. Facilitation. All right, this condition is where we have a particular neuron in a pool that could receive, that may receive, excitatory or inhibitory stimulation. If the overall effect, the net effect, is excitatory, but it lacks enough threshold potential there, then the neuron becomes more excitable to incoming stimulation. We term this facilitation. Another feature that we have to consider is convergence. You again can have a single neuron within a neuronal pool here that would receive impulses from two or more fibers. This would be convergence. This makes it possible for the neuron to summate impulses from different sources. And it continues, and sometimes we get things a little confused, but we have a pretty marvelous nervous system here. Divergence, again, going away from these leave a neuron in a pool. These impulses would, and they may be passed into several output fibers. They would diverge from one another. Here you can see examples of convergence, divergence. Let's talk about different types of nerves. A nerve is a bundle of nerve fibers that's held together in by layers of connective tissue. Nerves bring information to the central nervous system as sensory fibers, or we say bringing it to, these are afferent fibers. And we also have motor fibers, which are efferent. They carry impulses from the CNS, from our central system, the brain and spinal cord, out to the effectors, sometimes being muscles, glands, etc. So keep in mind, afferent and efferent. The routes that nerve impulses travel are called pathways, and the simplest is a reflex arc. A reflex arc includes a sensory receptor, a sensory neuron, an interneuron that we would find in the reflex center there in the CNS. Then you would have a motor neuron and an effector. Reflex arcs. Reflex behavior. Reflexes are automatic responses to stimuli in or out of the body. These will help us to maintain homeostasis. Keep in mind, homeostasis, maintenance of a stable internal environment. This can be like heart rate, blood pressure, whatever you may want to call it there. And these also, these reflexes will carry out automatic responses when we would have to sneeze, when we would vomit, swallowing. So there's a lot of different types of reflexes there. We have, we can cross our knee, we can hit right below the tendon right below our kneecap where it connects to our shin there. This is the patellar reflex. There is no interneuron involved there. This is a simple reflex, the knee-jerk reflex. Withdrawal reflex involves sensory neurons, interneurons, as well as motor neurons. We also have antagonistic extensor muscles would be inhibited there. Antagonistic, against, something that wouldn't allow it to happen. So, real good illustration here. You can follow the process of the mallet hitting, the patellar mallet hitting the ligament there below our kneecap. Contraction then of the quadriceps muscle group, and follow that back on around to the spinal cord, the way that it would come back, cause contraction, and the knee would, or the foot would jerk outward. Same way here with stepping on a tack or a pebble one, works the same way. Alright, let's talk about the meninges. The brain and the spinal cord are surrounded by membranes. Now keep in mind, the brain and spinal cord make up what? Central system. And these meninges lie between the bone and the soft tissue. We've got real good illustrations here, and I want you to pay very close attention to the detail of the skull there, the vertebral column, the brain. If you look there on slide A, part A there of slide 53, you'll see the meninges there at the bottom. They're in the vertebral column. You can see these little layers of membrane here. So this is the dura mater, the arachnoid matter, and the pia mater. Let's look at those. Keep in mind, dura mater means tough mother. The pia mater is little mother, and the arachnoid matter references a web-like feature. So if you'll follow the little square rectangle there at the top of the skull, come over and look at it in detail, you can see all of these layers through the brain. And notice that it's in one of those, it's picking out one of the convolutions here in the brain. And this is very important in it providing more surface area for these processes to take place. Keep in mind, in biological actions, surface area is very, very important. You can see all aspects. You can see the skin, subcu tissue. You can see the part of the skull, the bone there, all through the different meninges. You can see the subarachnoid space. You can see the web-like features crossing over there. You can see the gray matter and the white matter there that makes up the cerebrum. So, real good illustration. I can't emphasize that one enough. All right, outermost part of the meninges here is made up of tough, white, dense connective tissue, is very vascular, and is termed the tough mother, dura mater. It forms the inner periosteum. Periosteum is around the skull bone. That's the inner layer of that of the skull. In some areas, the dura mater, this tough mother, very dense, white connective tissue, very tough. It forms partition between the lobes in the brain, and it forms these dural sinuses. There's a sheath around the spinal cord that's separated from the vertebrae by an epidural space. So perhaps you've had an epidural. I've had several epidurals. Guys, maybe you haven't had an epidural yet, but it may be coming. And some of you have had children, you ladies, you will have probably had an epidural. All right, the middle meninx. This is the arachnoid matter. It is very thin, it lacks blood vessels, it does not follow the convolutions of the brain. It's between the arachnoid matter and the pia matter. This is called the subarachnoid space. And it contains cerebrospinal fluid. The innermost pia matter, the little matter, is very thin. It contains many blood vessels and many nerves. It's attached to the surface of the brain and the spinal cord, and it follows their contours. So go back and reference your illustration of your skull and the brain, and you can see all of that. You may have a picture there in your book that you can follow along with. All right, slide 56. Here again, you can see all of these. You can see the spinal cord inside the pia mater. You can see the arachnoid matter and the dura mater, all in these vertebrae. And then you can look at a cross section there in slide B there on slide 56 there in part B. All right, real good illustration. All right, spinal cord begins at the base of the brain, extends as a slender cord to the level of the intervertebral disc that we would find between the first and second lumbar vertebrae. First and second lumbar. Spinal cord. All right, you might need to examine that a little bit. Real good illustrations here. You can see the cervical enlargement. You can see the lumbar enlargement. You can see the spinal cord. You can see what would be termed the vertebral canal there. Keep in mind that This spinal cord can also be turned, and it's very descriptive here. I like to use it. This is what my surgeon uses. He says the spinal cord is a sack of nerves. Aptly put, very descriptive. I like it. All right, spinal cord consists of 31 segments, each of which gives rise to a pair of spinal nerves. And I mentioned the cervical enlargement gives rise to the nerves leading to the upper limbs, and a lumbar enlargement gives rise to those innervating the lower limbs. So you have the anterior medial fissure and the posterior medial fissure. This divides, these longitudinal grooves, divides the cord into right and left halves. All right, white matter, we've heard of that. This is made up of bundles of myelinated nerve tracts. It has these wrappings on it. It is myelinated. And it surrounds a butterfly-shaped core of gray matter that will house the inner neurons. The upper and lower wings are the posterior and anterior horns of the gray matter. Between them is the lateral horn. All right. The gray matter divides the white matter into three regions: anterior, lateral, and posterior funiculi. Each funiculus consists of longitudinal tracts, and you have that central canal that will contain CSF. All right, real good illustration. One is a photomicrograph, and the other one is an illustration. All of that is labeled. You can see where you have the central canal there. You can see the posterior funiculus. You can see the posterior horn. Look at the anterior funiculus there and the anterior horn. You can see the ventral root of a spinal nerve. You can see how the spinal nerves on the left there of A branch off. So you've got a lot of good illustrations here. I encourage you to look at those and read more about it in your text. All right, functions of spinal cord. Two major functions: to transmit impulses to and from the brain, as well as to house spinal reflexes. We have tracts carrying sensory information, these electrochemical impulses, to the brain, and if they're carrying it to the brain, they're ascending tracts, and if they're carrying it from the brain, we term that as descending tracts that's carrying this electrochemical information. The names that identify the nerve tracts identify the origin and the termination of the fibers within that tract, and there's a lot of spinal reflexes passing through the spinal cord as well. Another good illustration. I encourage you to follow that. It's pretty self-explanatory, and your text may give you some more good information on it there. Same way on that slide, 64. All right, the brain. Brain is the largest, the most complex portion of the nervous system. I can't fathom 100 billion of anything. We have 100 billion multipolar neurons within our brain. It is very complex. It can be divided into the cerebrum. That's the largest portion. That's where most of our higher mental functions occur. We have the diencephalon. This processes sensory input, and we have the cerebellum at the back of our brain, so to speak, and that coordinates muscular activity. Then we have the brain stem that will take care of our visceral activities, coordinates and regulates visceral activities. Real good illustration here again. You can see all of these features. I encourage you to study that along with your text. Cerebrum consists of two cerebral hemispheres. You have a right hemisphere and a left hemisphere. You have the ridge that goes down the center there of nerve fibers. That's called the corpus callosum. It connects the two hemispheres, and then the brain has these different features. the gyri, the sulci, and the fissures, them being longitudinal or transverse. Lobes of the brain are named according to the bones they lie under and include the frontal lobe, the parietal lobe, the temporal lobe, the occipital lobe, and the insula. There's a thin layer of gray matter that we term the cerebral cortex. It lies on the outside of the cerebrum now. Where's the cerebrum? Yeah. It contains 75% of the cell bodies in the nervous system. Wow. Beneath the cortex lies a mass of white matter that's made up of myelinated nerve fibers. These connect the cell bodies of the cerebral cortex with the rest of the nervous system. Myelinated, mass of white matter, myelinated nerve fibers. All right, they're going to be controlled, going to be contained. They are directed. Functions of the cerebrum. It provides higher brain functions, interpretation of sensory input, initiating voluntary muscle movements, memory, integrating information for reasoning. We have functional regions in the cerebral cortex. The functional areas of the brain overlap, but the cortex can generally be divided into motor, sensory, and association areas. The primary motor areas lie in the frontal lobes, anterior to the central sulcus, and in its interior wall. This region includes the parietal cells. The sensory areas are located in several areas of the cerebrum. They interpret sensory information, they produce feelings, they produce sensations. We have these association areas in the brain that analyze and interpret sensory impulses, helps us to reason, it gives us our judgment, helps us to make decisions about our emotions perhaps, or verbalizing ideas, as well as storing memory. Sensory areas for sight lie within the occipital lobe, and sensory and motor fibers will cross over in the spinal cord and the brainstem, so that centers in the right hemisphere are interpreting or controlling the left side of the body, and vice versa. We have association areas of the frontal lobe, control a number of higher intellectual processes. A general interpretive area is found at the junction of the parietal, temporal, and occipital lobes, and this plays a primary role in very complex thought processing. Good illustration here. You can see what is taking place in different areas of your brain. I encourage you to look at that carefully, see how all that will function. And I believe we'll get into more of this in a different section. We're finished with this one, and I will stop here and pick up later on in chapter 9.